Hello, we're continuing with life science and cell biology in a nutshell. We're learning about the steps of cell division, or the phases. So we learned that G1, gap phase 1, is the very beginning of cell division. So the cell increases in mass in preparation for cell division. Right after G1, you have the S phase, which is the synthesis phase where DNA is synthesized. And G2 is the gap 2 phase, right before prophase begins. And again, the proteins, more proteins are made, and the cell continues to increase in size. And the nucleoli are still present at this stage. So the cell chromosomes have already duplicated, and due to S phase, and they're still bound in the nuclear envelope of the chromosomes. You have two pairs of centrioles, which are located outside of the nucleus. So one pair has replicated into two pairs of these protein cylindrical fiber structures. So in early prophase, chromatin are coiled into chromosomes with chromatids. Each of the two chromatids, or each strand of the chromosomes, joined at a centromere in the center. The mitotic spindle forms in the cytoplasm, and you have asters, which are the fibers around each centriole pair. So these are this is as far as the structure and the fibers are concerned. So microtubules lengthen. They the two pairs of centrioles are pulled apart to opposite ends of the cell. All the fibers lengthen. And in late prophase, the nuclear envelope finally breaks up. Polar fibers, which are microtubules making up the spindle fibers, reach from each cell pole to the cell's equator. Kinetochores, which are specified regions in the centromeres of chromosomes, attach to a type of microtubule called kinetochore fibers. The kinetochore fibers interact with the spindle polar fibers, connecting the kinetochores to the polar fibers. Chromosomes begin to migrate towards the cell center. In metaphase, the spindle fully develops and the chromosomes align at the metaphase plate, a plane that is equally distant from the two spindle poles. And the nuclear membrane disappears completely. In animal cells, the two pair of centrioles align at opposite poles of the cell. Polar fibers, microtubules making up the spindle fibers, continue to extend from the poles to the center of the cell. Chromosomes move randomly until they attach at their kinetochores to polar fibers from both sides of their centromeres. Chromosomes align at metaphase plate at right angles to the spindle poles, and they're held at the metaphase plate by equal forces of the polar fibers pushing on the centromeres of the chromosomes. So this is what it looks like, the steps, and then the lining up. That's the asters, and these are in metaphase, the genetic material. And then in anaphase, the paired chromosomes or sister chromatids separate, moving to opposite ends or poles of the cell. Spindle fibers are not Spindle fibers that are not connected to chromatids lengthen and elongate the cell. At the end of anaphase, each pole contains a complete compilation of chromosomes. So they have migrated to opposite ends, the chromosomes. The paired centromeres in each distinct chromosome begin to move apart. Once the paired sister chromatids separate from one another, each is considered a full chromosome. They are referred to as daughter chromosomes. Through the spindle apparatus, the daughter chromosomes move to the poles at opposite ends of the cell. The daughter chromosomes migrate centromere first, and the kinetochore fibers become shorter as the chromosomes are nearing the pole. In preparation for telophase, the two cell poles also move further apart during the course of anaphase. At the end of anaphase, each pole contains a complete compilation of chromosomes. Cytokinesis, the division of the original cell cytoplasm, begins and continues through telophase. In telophase, the polar fibers continue to lengthen. Nuclei begin to form at opposite poles. The nuclear envelopes of these nuclei are formed from remnant pieces of the parent cell's nuclear envelope and from pieces of the endomembrane system. Nucleoli also they reappear, which are inside the nucleus. The part where you have a protein subunits of ribosomes form. Chromatin fibers of chromosomes uncoil. After these changes, telophase and mitosis is pretty much complete. Genetic contents of one cell have been divided equally into two, and cytokinesis, which begins in anaphase, completes shortly after telophase. So during these phases, chromosomes are separated, moving to opposite poles of the cell. Now they're newly formed nuclei. At the end of the division process, duplicated chromosomes are equally divided between the two cells. The daughter cells are genetically identical diploid cells that have the same chromosome number and chromosome type as the parent cells. Somatic cells are examples of cells that divide by mitosis. Somatic cells consist of all body cell types, excluding sex cells. The somatic cell chromosome number in humans is 46, while the chromosome number for sex cells is 23. In organisms that are capable of sexual reproduction, daughter cells are produced by meiosis. Meiosis is a two-part division process producing gametes. Gametes are the sex cells. The dividing cell goes through mainly the same steps as in mitosis. So prophase, metaphase, anaphase, and then telophase. It goes through twice actually for meiosis. At the end of meiosis and cytokinesis, 4 haploid cells instead of diploid are produced from a single diploid cell. So in a sense, it's actually in a way the opposite of mitosis, because, well, not exactly opposite, but instead of having a cell number multiply by 2, it's actually dividing by 2 in a way. So these haploid daughter cells have half the number of chromosomes as the parent cell and are not genetically identical to the parent cell. So we'll continue in the next part learning more about this type of division. Thank you.